This is a summary of how to control hunger, eating, and satiety from Hubermanlab Essentials, where we break down the key insights, ideas, and takeaways from this content, giving you the gist so you can decide if you want the rest. Introduction. In this Hubermanlab Essentials episode, Dr. Andrew Huberman, a professor of neurobiology and ophthalmology at Stanford School of Medicine, explores the complex interplay between hormones, the nervous system, and eating behaviors. He provides practical, science-based tools and insights into how hormones regulate hunger, appetite, and feelings of fullness, satiety. Dr. Huberman clearly explains the hormonal and neurological mechanisms involved, highlighting specific practices to help listeners effectively manage eating behaviors, maintain healthy body composition, and improve overall metabolic health. The discussion explores how specific hormones, including ghrelin, melanocyte-stimulating hormone MSH, cholecystokinin, CCK, insulin, and glucagon, regulate hunger, appetite, and satiety. The critical roles of the hypothalamus, particularly the ventromedial hypothalamus and arcuate nucleus, and the insular cortex in managing eating behaviors, hunger cues, and sensory responses to food. How ghrelin secretion acts like a hunger clock, creating anticipatory signals based on habitual eating times, explaining why regular meal timing significantly affects hunger sensations. The impact of melanocyte-stimulating hormone, MSH, on reducing appetite and the unique role sunlight exposure plays in increasing MSH secretion. The specific nutrients, omega-3 fatty acids, conjugated linoleic acid, CLA, and glutamine, that stimulate the secretion of the hormone cholecystokinin, CCK, significantly reducing appetite. How highly processed foods containing emulsifiers damage the gut lining, preventing proper appetite regulation, increasing overeating, and negatively impacting metabolic health. The critical roles insulin and glucagon play in blood sugar regulation, energy management, and the importance of meal composition and timing for stable glucose levels. Practical dietary strategies for managing blood sugar and appetite, such as nutrient order during meals and strategic use of movement to stabilize blood glucose. The role and impact of supplements and substances like metformin, berberine, and yerba mate in managing appetite, glucose levels, and overall metabolic health. Deep dive into main ideas. Dr. Huberman emphasizes the brain's central role in controlling hunger and satiety, highlighting two critical brain areas involved: the ventromedial hypothalamus and the arcuate nucleus. The arcuate nucleus contains two crucial neuron populations: AGRP neurons, which promote hunger, and POMC neurons, producing melanocyte-stimulating hormone, MSH, significantly reducing hunger. MSH, stimulated notably by exposure to sunlight, potently suppresses appetite, offering practical insights into controlling hunger through behavioral lifestyle practices like consistent sunlight exposure. He explains ghrelin, a hormone secreted from the gut primarily when glucose levels drop, creating sensations of hunger and food anticipation based on regular meal timing. Regular eating times set a ghrelin-driven hormonal clock, making hunger predictable. If eating patterns shift abruptly, ghrelin's anticipatory release still occurs, creating intense cravings. Huberman's practical advice includes gradually shifting meal times to adapt this hormonal clock, effectively minimizing hunger discomfort during dietary changes. Another hormone discussed extensively is cholecystokinin, CCK, released from the gut in response to certain dietary nutrients. CCK significantly reduces appetite, specifically triggered by omega-3 fatty acids, found in fish and certain supplements, conjugated linoleic acid, CLA, and amino acids like glutamine. These nutrients, when consumed, enhance satiety, reducing overall food intake naturally. This underscores the practical importance of dietary choices rich in these nutrients to manage hunger and achieve satiety effectively. Huberman clearly explains how highly processed foods negatively impact appetite control. Specifically, emulsifiers common in processed foods damage the gut lining and disrupt gut-brain signals critical for satiety, such as those triggered by CCK. By stripping the mucosal lining and harming gut neurons, processed foods prevent the body from accurately sensing nutrient intake, driving overeating. Simultaneously, hidden sugars in these foods trigger excessive dopamine release, fueling compulsive eating. Huberman strongly advises consuming whole, minimally processed foods to preserve gut health and natural appetite regulation. The discussion emphasizes the importance of insulin in blood glucose management, vital for metabolic health. Eating meals consisting of carbohydrates, fats, and proteins raises glucose and insulin levels, but the order and timing significantly impact blood glucose spikes. Eating fiber-rich foods first, followed by proteins and carbohydrates, can effectively moderate blood sugar increases, preventing rapid glucose spikes and crashes that lead to overeating. Practical movement strategies are offered for optimal glucose management. Huberman recommends moderate exercise or simple movement like walking shortly after meals, significantly stabilizing glucose levels, improving insulin sensitivity, and preventing blood sugar fluctuations that could lead to overeating. He explains this simple yet powerful strategy provides both immediate and long-term metabolic health benefits. Huberman addresses pharmacological interventions for glucose management, such as metformin, highlighting its powerful ability to lower blood sugar by affecting liver mitochondria and enhancing insulin sensitivity. He emphasizes the significant benefits, but also cautions around careful use under medical supervision, as these interventions are potent and carry potential risks. Another practical suggestion is yerba mate tea, which uniquely boosts hormones like glucagon-like peptide-1, GLP-1, helping regulate hunger effectively, alongside improving alertness and cognitive performance. Huberman personally incorporates yerba mate strategically into his routine, leveraging its dual effects on mental clarity and hunger control. Finally, Huberman highlights the critical role of electrolytes for optimal neurological function, cognitive clarity, and metabolism. Electrolytes, sodium, potassium, and magnesium, are essential for neuronal function, with proper electrolyte balance significantly enhancing performance, reducing brain fog, and supporting overall health. Huberman underscores practical hydration strategies, including using electrolyte-rich beverages to optimize cognitive and physical performance throughout the day. Conclusion: understanding how hormones regulate hunger, eating, and satiety provides powerful practical tools to enhance health, cognitive performance, and quality of life. Dr. Huberman offers clear, scientifically validated insights and actionable strategies, including meal composition, timing adjustments, dietary supplementation, and behavioral techniques, empowering listeners to effectively manage appetite, enhance metabolic health, and optimize their overall well-being. That's a wrap on how to control hunger, eating, and satiety. If you found this summary valuable, make sure to follow, subscribe, and come back for more in-depth breakdowns of content.